A virus is a fascinating and complex entity that straddles the line between living and non-living things. Let's break down its characteristics and functions in detail. 1. Definition and basic structure. A virus is a microscopic infectious agent that can only replicate inside the living cells of an organism. It consists of nucleic acid core, this can be either DNA or RNA, which contains the genetic information necessary for the virus to reproduce. Protein coat, capsid, this surrounds the nucleic acid and protects it. The capsid is made up of protein subunits called capsomes. Envelope, some viruses have an additional lipid envelope derived from the host cell membrane, which can contain viral proteins that help the virus infect new cells. 12. 2. Replication process. Viruses cannot reproduce on their own. They must infect a host cell and hijack its machinery to replicate. The replication process typically involves several steps. Attachment, the virus binds to specific receptors on the surface of the host cell. Penetration, the virus or its genetic material enters the host cell. Uncoating, the viral capsid is removed, releasing the viral nucleic acid into the host cell. Replication and transcription, the host cell's machinery is used to replicate the viral genome and produce viral proteins. Assembly, new viral particles are assembled from the replicated genetic material and proteins. Release, new viruses are released from the host cell, often destroying it in the process, to infect other cells. 12. 3. Types of viruses. Viruses can be classified based on several criteria, including their shape, type of nucleic acid, and mode of replication. Some common types include bacteriophages, viruses that infect bacteria; retroviruses, viruses that use reverse transcription to convert their RNA into DNA inside a host cell, for example, HIV. DNA viruses, viruses that have DNA as their genetic material, for example, herpes virus. RNA viruses, viruses that have RNA as their genetic material, for example, influenza virus. 12. 4. Impact on hosts. Viruses can cause a wide range of diseases in humans, animals, and plants. Some well-known viral diseases include HIV/AIDS, caused by the human immunodeficiency virus, HIV. COVID-19, caused by the SARS-CoV-2 virus. Influenza, caused by various strains of the influenza virus. Measles, caused by the morbillivirus 2. 5. Evolution and adaptation. Viruses evolve rapidly due to their high mutation rates and short generation times. This allows them to adapt quickly to new hosts and environments, often leading to the emergence of new viral strains and outbreaks. 1. 6. Role in research and biotechnology. Viruses are not only pathogens but also valuable tools in scientific research and biotechnology. They are used in gene therapy. Modified viruses can deliver therapeutic genes to treat genetic disorders. Vaccines. Some vaccines use weakened or inactivated viruses to stimulate an immune response without causing disease. Molecular biology. Viruses are used to study fundamental biological processes and gene function. 1. Understanding viruses is crucial for developing treatments and preventive measures against viral diseases. Their unique properties make them both a challenge and a tool in the field of biology and medicine. Viruses mutate through a process that involves changes in their genetic material. These mutations can occur due to errors during the replication of their genetic material, which can be either DNA or RNA. Here's a detailed breakdown of how this happens. 1. Replication errors. When a virus infects a host cell, it hijacks the cell's machinery to replicate its genetic material and produce new viral particles. During this replication process, errors can occur, leading to mutations. These errors are more common in RNA viruses than in DNA viruses because RNA-dependent RNA polymerases, the enzymes that replicate RNA, lack proofreading mechanisms. 12. 2. Types of mutations. Mutations can be of various types, including point mutations, a single nucleotide change in the viral genome; insertions and deletions, addition or loss of nucleotides in the genome; reassortment, exchange of genetic material between different viral strains, common in segmented RNA viruses like influenza. 12. 3. Consequences of mutations. The effects of mutations can vary. Neutral mutations, these do not significantly affect the virus's properties. Beneficial mutations, these can enhance the virus's ability to infect hosts, evade the immune system, or increase transmissibility. Deleterious mutations, these can impair the virus's ability to replicate or infect hosts. 12. For evolution and selection. Mutations that provide a survival advantage are more likely to be passed on to future generations of the virus. This process of natural selection can lead to the emergence of new viral strains or variants that are better adapted to their environment. 12. 5. Impact on public health. Mutations can have significant implications for public health. Vaccine efficacy. Some mutations can alter viral proteins targeted by vaccines, potentially reducing vaccine effectiveness. Antiviral resistance. Mutations can lead to resistance against antiviral drugs. Epidemiology. New variants can change the dynamics of viral transmission and disease severity. 12. 6. Examples of viral mutation. Influenza virus, known for its high mutation rate, leading to seasonal flu variations and the need for annual vaccine updates. SARS-CoV-2, the virus responsible for COVID-19, has undergone numerous mutations, resulting in variants like Alpha, Delta, and Omicron, each with different transmissibility and immune escape properties. 12. Understanding how viruses mutate helps scientists develop better strategies for controlling viral infections, including designing effective vaccines and antiviral therapies. Mutations are changes in the genetic material of an organism, and they can occur in various forms. Here are the most common types of mutations. 1. Mutations. These involve changes to a single nucleotide base in the DNA sequence. They can be further classified into silent mutations. These do not change the amino acid sequence of the protein. Missense mutations. These result in the substitution of one amino acid for another in the protein. Nonsense mutations. These introduce a premature stop codon, leading to a truncated and usually non-functional protein. 12. 2. Insertions and deletions, indels. These mutations involve the addition or removal of one or more nucleotide bases in the DNA sequence. They can cause frameshift mutations. When the number of inserted or deleted bases is not a multiple of 3, it shifts the reading frame of the gene, often resulting in a completely different and usually non-functional protein. 12. 3. Duplication. A segment of the DNA is copied one or more times. This can lead to an increase in the dosage of the genes within the duplicated segment, potentially affecting the organism's phenotype. 12. 4. Inversions. A segment of the DNA is reversed within the chromosome. This can disrupt the function of genes at the breakpoints and affect gene regulation. 12. 5. Translocations. A segment of DNA is moved from one location to another, either within the same chromosome or to a different chromosome. This can lead to gene fusion or misregulation of genes at the breakpoints. 12. 6. Large-scale mutations. These involve larger segments of DNA and can include chromosomal deletions, loss of a large segment of a chromosome; chromosomal duplications, duplication of a large segment of a chromosome; chromosomal inversions, reversal of a large segment within a chromosome; chromosomal translocations, transfer of a large segment from one chromosome to another. 12. 7. Germline versus somatic mutations. Germline mutations occur in the reproductive cells and can be passed on to offspring. Somatic mutations occur in non-reproductive cells and are not passed on to offspring but can lead to diseases like cancer. 12. Understanding these types of mutations is crucial for studying genetic diseases, evolution, and the development of new therapies. Large-scale chromosomal deletions can have profound and varied consequences, depending on the size of the deletion and the genes affected. Here are some key points to consider. 1. Loss of gene function. When large segments of a chromosome are deleted, multiple genes can be lost. This can lead to loss of essential genes. If the deleted region includes genes essential for survival, it can result in severe developmental issues or lethality. Haploinsufficiency. This occurs when a single copy of a gene is not sufficient to maintain normal function, leading to disorders. 12. 2. Genetic disorders. Large-scale deletions are often associated with various genetic disorders. Some examples include Cruduchat syndrome, caused by a deletion on chromosome 5, leading to intellectual disability and a characteristic cat-like cry in infants. DI-George syndrome, resulting from a deletion on chromosome 22. It can cause heart defects, immune deficiencies, and developmental delays. 34. 3. Cancer development. Chromosomal deletions can contribute to cancer by removing tumor suppressor genes, which normally help control cell growth and division. The loss of these genes can lead to uncontrolled cell proliferation and tumor formation. 12. 4. Metabolic and physiological changes. Deletions can disrupt metabolic pathways and physiological processes by removing genes involved in these functions. This can lead to metabolic disorders, disruption of enzymes and other proteins involved in metabolism, physiological abnormalities, changes in hormone levels, immune function, and other bodily processes. 45. 5. Cellular heterogeneity and adaptation. In some cases, large-scale deletions can lead to cellular heterogeneity, where different cells within the same organism have different genetic compositions. This can result in premature senescence, accelerated aging of cells, drug resistance. In cancer, this heterogeneity can lead to resistance to chemotherapy and other treatments. 45. 6. Evolutionary impact. While often deleterious, large-scale deletions can also play a role in evolution by creating genetic diversity. In some cases, they may provide a selective advantage under certain environmental conditions. 45. Understanding the consequences of large-scale chromosomal deletions is crucial for diagnosing and treating genetic disorders, as well as for developing targeted therapies in cancer treatment. Loss of tumor suppressor genes. Tumor suppressor genes are crucial for regulating cell growth and preventing uncontrolled cell division. When a chromosomal deletion removes one or more of these genes, it can lead to loss of function. Without these genes, cells can grow uncontrollably, leading to tumor formation. Haploinsufficiency. Sometimes, having only one functional copy of a tumor suppressor gene, instead of the usual two, is not enough to maintain normal function, which can also promote cancer. 12. 2. Genomic instability. Deletions can cause genomic instability, which is a hallmark of cancer. This instability can lead to further mutations. The initial deletion can make the genome more susceptible to additional mutations, amplifications, and other chromosomal abnormalities. Chromosomal rearrangements. These can create fusion genes or disrupt normal gene function, contributing to cancer progression. 12. 3. Disruption of regulatory regions. Chromosomal deletions can remove regulatory regions that control the expression of genes. This can lead to misregulation of gene expression. Genes that are normally tightly regulated can become overexpressed or underexpressed, contributing to cancer development. 12. 4. Impact on cell cycle and apoptosis. Deletions can affect genes involved in the cell cycle and apoptosis, programmed cell death. This can result in uncontrolled cell division. Loss of genes that regulate the cell cycle can lead to unchecked cell proliferation. Resistance to cell death. Deletions of genes involved in apoptosis can make cancer cells resistant to programmed cell death, allowing them to survive and proliferate. 12. 5. Examples of cancer-related deletions. Retinoblastoma. Deletions in the RB1 gene on chromosome 13 can lead to retinoblastoma, a type of eye cancer. Breast cancer. Deletions in the BRCA1 or BRCA2 genes increase the risk of breast and ovarian cancers. Colorectal cancer. Deletions in the APC gene on chromosome 5 are associated with familial adenomatous polyposis, a condition that can lead to colorectal cancer. 12. 6. Therapeutic implications. Understanding the role of deletions in cancer can help in developing targeted therapies, gene therapy, strategies to replace or repair deleted genes. Targeted drugs. Drugs that specifically target pathways affected by the deletions. Personalized medicine, tailoring treatments based on the specific genetic deletions present in a patient's tumor. 12. Chromosomal deletions are a critical factor in the development and progression of many cancers. By studying these deletions, researchers can better understand cancer mechanisms and develop more effective treatments. Viruses have evolved various sophisticated mechanisms to evade the immune system, allowing them to persist and replicate within their hosts. Here's a detailed breakdown of these strategies. 1. Antigenic variation. Viruses can change the proteins on their surface to avoid detection by the immune system. This process, known as antigenic variation, involves mutation, rapid changes in the viral genome, especially in RNA viruses like influenza and HIV, can alter surface proteins so that antibodies generated against previous infections or vaccinations no longer recognize the virus. 1. Recombination and reassortment. Exchange of genetic material between different viral strains can create new variants with different surface antigens. This is common in viruses with segmented genomes, such as the influenza virus. 1. 2. Inhibition of antigen presentation. Viruses can interfere with the host's ability to present viral antigens to immune cells. Downregulation of MHC molecules. Some viruses, like cytomegalovirus (CMV), can reduce the expression of major histocompatibility complex (MHC) molecules on the surface of infected cells. This prevents the immune system from recognizing and destroying these cells. 2. Interference with antigen processing. Viruses can produce proteins that inhibit the processing of viral antigens within the host cell, further preventing their presentation on MHC molecules. 2. 3. Evasion of antibody responses. Viruses can evade neutralizing antibodies through several mechanisms. Glycan shielding. Some viruses, like HIV, cover their surface proteins with sugar molecules, glycans, that shield them from antibody recognition. 3. Conformational masking. Viruses can hide critical epitopes, parts of antigens recognized by antibodies, by changing the conformation of their surface proteins. 3. 4. Latency and integration. Certain viruses can enter a latent state, where they remain dormant within host cells and evade immune detection. Latency. Herpes viruses, such as Epstein-Barr virus, EBV, and herpes simplex virus, HSV, can establish latency in host cells, where they do not produce viral proteins that could be detected by the immune system. 4. Integration. Retroviruses like HIV integrate their genetic material into the host genome, allowing them to persist in a latent form and reactivate later. 4. 5. Modulation of host immune responses. Viruses can manipulate the host immune response to their advantage. Cytokine mimicry and modulation. Some viruses produce proteins that mimic host cytokines, immune signaling molecules, or interfere with cytokine signaling, dampening the immune response. Inhibition of apoptosis. Viruses can produce proteins that inhibit programmed cell death, apoptosis, of infected cells, allowing them to survive and replicate longer. 6. Escape from cellular immune responses. Viruses can evade detection and destruction by cytotoxic T lymphocytes, CTLs. Mutations in CTL epitopes. Mutations in viral proteins can alter CTL epitopes, preventing recognition by CTLs. Inhibition of CTL function. Some viruses produce proteins that directly inhibit CTL activity or induce the expression of inhibitory molecules on the surface of infected cells. 7. Subversion of innate immune responses. Viruses can also evade the innate immune system, which provides the first line of defense. Inhibition of interferon responses. Many viruses produce proteins that inhibit the production or signaling of interferons, which are crucial for antiviral defense. Blocking pattern recognition receptors, PRs. Viruses can interfere with PRs, which detect viral components and trigger immune responses. Understanding these evasion strategies is crucial for developing effective antiviral therapies and vaccines. By targeting these mechanisms, researchers can design interventions that enhance the immune system's ability to detect and eliminate viruses. Viruses have evolved numerous strategies to manipulate host cytokines, which are crucial signaling molecules in the immune system. Here's a detailed breakdown of how viruses achieve this. 1. Cytokine mimicry. Some viruses produce viral proteins that mimic host cytokines. These viral cytokines can bind to host cytokine receptors and modulate immune responses. Viral interleukin 10, VIL-10. Certain viruses, like Epstein-Barr virus, EBV, produce VIL-10, which mimics the host's IL-10. This cytokine suppresses the immune response by inhibiting the activity of T cells and macrophages, helping the virus evade detection. 1. 2. Cytokine receptor mimicry. Viruses can also produce proteins that mimic cytokine receptors, which can bind to host cytokines and neutralize their effects. Viral chemokine receptors. Some viruses, such as cytomegalovirus, CMV, encode viral chemokine receptors that can bind to host chemokines, preventing them from recruiting immune cells to the site of infection. 2. 3. Inhibition of cytokine production. Viruses can interfere with the host's ability to produce cytokines, thereby dampening the immune response. Inhibition of interferon production. Many viruses, including influenza and hepatitis C virus, HCV, produce proteins that inhibit the production of interferons, which are critical for antiviral defense. 3. 4. Modulation of cytokine signaling pathways. Viruses can manipulate the signaling pathways that are activated by cytokines to alter the immune response. Interference with JAK-STAT pathway. The Janus kinase signal transducer and activator of transcription. JAK-STAT pathway is a key signaling mechanism for many cytokines. Viruses like Vaccinia virus produce proteins that inhibit this pathway, reducing the effectiveness of cytokine signaling. 4. 5. Induction of anti-inflammatory cytokines. Some viruses can induce the production of anti-inflammatory cytokines to create a more favorable environment for their replication. Induction of IL-10 and TGF. By promoting the production of anti-inflammatory cytokines like IL-10 and transforming growth factor beta, TGF, viruses can suppress the immune response and avoid detection. 5. 6. Exploitation of cytokine networks. Viruses can exploit the complex networks of cytokine interactions to their advantage. Cytokine storms. In some cases, viruses can trigger an excessive immune response known as a cytokine storm. This can cause severe tissue damage and help the virus spread by disrupting normal immune function. SARS-CoV-2, the virus responsible for COVID-19, is known to induce cytokine storms in severe cases. 6. 7. Blocking cytokine receptors. Viruses can produce proteins that block host cytokine receptors, preventing cytokines from binding and signaling. Viral IL-1 receptor antagonist. Some viruses produce proteins that act as antagonists to IL-1 receptors, blocking the pro-inflammatory effects of IL-1 and reducing the immune response. 8. Sequestration of cytokines. Viruses can sequester cytokines to prevent them from reaching their target cells. Binding proteins. Certain viruses produce binding proteins that capture and sequester cytokines, preventing them from activating immune cells. Understanding these mechanisms is crucial for developing antiviral therapies and vaccines that can effectively counteract viral evasion strategies. By targeting these viral manipulations, researchers can enhance the immune system's ability to detect and eliminate viruses. Cytokine storms are a critical and often deadly aspect of severe COVID-19 cases. Here's a detailed explanation of what they are, how they occur, and their implications. 1. Definition of cytokine storm. A cytokine storm, also known as cytokine release syndrome (CRS), is an excessive and uncontrolled release of pro-inflammatory cytokines. This hyperactive immune response can lead to severe inflammation and tissue damage. 12. 2. Mechanism in COVID-19. In COVID-19, the cytokine storm is triggered by the body's immune response to the SARS-CoV-2 virus. Here's how it typically unfolds. Initial infection. The virus infects cells in the respiratory tract, particularly the lungs. Immune activation. The immune system detects the virus and activates immune cells, such as macrophages and T helper cells, which release cytokines to recruit more immune cells to the site of infection. Excessive cytokine production. In some patients, this response becomes dysregulated, leading to an overproduction of cytokines like interleukin-6, IL-6, interleukin-1, IL-1, tumor necrosis factor alpha, TNF, and others. 12. 3. Consequences of cytokine storm. The excessive cytokine release can have several severe consequences. Acute respiratory distress syndrome, ARDS. The inflammation can cause fluid buildup in the lungs, leading to ARDS, which is a major cause of death in COVID-19 patients. 12. Multi-organ failure. The systemic inflammation can affect multiple organs, including the heart, liver, and kidneys, leading to multi-organ failure. 12. Hypercoagulability. Increased cytokine levels can lead to abnormal blood clotting, contributing to complications like deep vein thrombosis and pulmonary embolism. 12. 4. Clinical features. Patients experiencing a cytokine storm may exhibit high fever, persistent high fever due to the inflammatory response, severe fatigue, extreme tiredness and weakness, respiratory distress, difficulty breathing and low oxygen levels, organ dysfunction, signs of organ failure, such as altered mental status, decreased urine output, and jaundice. 12. 5. A diagnosis. Diagnosing a cytokine storm involves clinical assessment, evaluating symptoms and signs of severe inflammation. Laboratory tests, measuring levels of inflammatory markers like C-reactive protein, CRP, ferritin, D-dimer, and specific cytokines, for example, IL-6, 12. 6. Treatment strategies. Managing cytokine storms in COVID-19 involves several approaches. Anti-inflammatory drugs, medications like corticosteroids, for example, dexamethasone, can reduce inflammation. Cytokine inhibitors, drugs that specifically target cytokines, such as tocilizumab, an IL-6 inhibitor, can help control the immune response. 12. Supportive care, providing oxygen therapy, mechanical ventilation, and other supportive measures to manage symptoms and organ dysfunction. 12. 7. Research and future directions. Ongoing research aims to better understand cytokine storms and develop more effective treatments. Biomarkers, identifying biomarkers to predict which patients are at risk of developing a cytokine storm. New therapies, developing new drugs and therapeutic strategies to modulate the immune response without compromising the body's ability to fight the virus. 12. Understanding cytokine storms is crucial for improving outcomes in severe COVID-19 cases. By targeting the underlying mechanisms, healthcare providers can better manage this life-threatening complication. The emergence of viruses is a complex and fascinating topic that intertwines with the history of life itself. Here's a detailed explanation of how viruses might have emerged, developing each point thoroughly. 1. Theories of viral origin. There are several hypotheses about how viruses originated, each with its own supporting evidence and implications. A regressive hypothesis, devolution. This hypothesis suggests that viruses may have evolved from more complex, free-living organisms that gradually lost the ability to survive independently. Parasitic ancestors. Viruses could have originated from parasitic organisms that became increasingly dependent on their hosts, eventually losing all cellular structures except for the genetic material and a protein coat. 1. Support. This theory is supported by the existence of large viruses, such as mimivirus, which possess genes typically found in cellular organisms. 1. B. Progressive hypothesis, escape. This hypothesis proposes that viruses originated from genetic elements that gained the ability to move between cells. Mobile genetic elements. Viruses may have evolved from plasmids or transposons, which are small DNA molecules within cells that can replicate independently and move between different cells. 2. Support. The similarity between some viral genes and those found in plasmids and transposons supports this hypothesis. 2. C. Virus first hypothesis. This hypothesis posits that viruses predate or co-evolved with the first cellular life forms. Precellular life. Viruses might have originated from self-replicating molecules in the probiotic world, before the emergence of cellular life. 3. Support. The simplicity of viral structures and their ability to replicate using host machinery suggest they could be remnants of an ancient precellular world. 3. 2. Evolutionary pathways. Viruses have followed diverse evolutionary pathways, leading to the vast array of viral forms we see today, at divergence and specialization. Genetic diversity. Viruses exhibit a wide range of genetic diversity, with genomes composed of DNA or RNA, single-stranded or double-stranded, and linear or circular forms. 4. Host specialization. Over time, viruses have evolved to infect specific hosts, ranging from bacteria, bacteriophages, to plants, animals, and humans. 4. B. Horizontal gene transfer. Gene exchange. Viruses can acquire genes from their hosts through horizontal gene transfer, which can enhance their ability to infect and replicate. 5. Genetic mosaicism. This process has led to the creation of viral genomes that are mosaics of genes from various sources, contributing to their adaptability and evolution. 5. 3. Impact on host evolution. Viruses have played a significant role in the evolution of their hosts, a genetic innovation. Gene transfer. Viruses can transfer genes between different species, promoting genetic diversity and innovation. Endogenous retroviruses. Some viral genes have been integrated into the genomes of their hosts, contributing to the evolution of new traits and functions. B. Selective pressure. Immune system evolution. The constant arms race between viruses and their hosts has driven the evolution of sophisticated immune systems. Population dynamics. Viral infections can influence population dynamics by affecting the survival and reproduction of their hosts. 4. Modern implications. Understanding the origins and evolution of viruses has important implications for modern science and medicine, and vaccine development. Targeting viral evolution. Knowledge of viral evolution helps in designing vaccines that can anticipate and counteract viral mutations. Broad-spectrum vaccines. Research into the common features of viruses can lead to the development of broad-spectrum vaccines that protect against multiple viral strains. B. Gene therapy. Viral vectors. Viruses are used as vectors in gene therapy to deliver therapeutic genes to specific cells. Safety and efficacy. Understanding viral mechanisms helps improve the safety and efficacy of these therapies. 5. Future research directions. Ongoing research aims to uncover more about the origins and evolution of viruses and ancient viral genomes. Paleovirology. Studying ancient viral genomes preserved in host DNA can provide insights into the long-term evolution of viruses. Viral fossils. Identifying viral sequences in ancient biological samples can help trace the history of viral infections. B. Synthetic virology. Creating synthetic viruses. Researchers are exploring the creation of synthetic viruses to study their properties and potential applications. Ethical considerations. This research raises important ethical questions about the creation and use of synthetic life forms. Understanding how viruses emerged and evolved is crucial for developing strategies to combat viral diseases and harnessing their potential for beneficial applications. Ancient viral genomes provide a fascinating glimpse into the history of viral evolution and their interactions with hosts. Here are some notable examples. 1. Human endogenous retroviruses, HERVs. HERVs are remnants of ancient retroviral infections that have integrated into the human genome. They make up about 8% of the human genome and have been passed down through generations. 1. Examples include HERV-K, HML2, one of the most recently active groups of HERVs, which went extinct less than 5 million years ago. Some HERV-K proviruses still retain the ability to produce viral proteins. 1. Syncytin, a gene derived from an ancient retrovirus that plays a crucial role in the formation of the placenta in mammals. 1. 2. Ancient viral DNA in glaciers. researchers have discovered ancient viral genomes preserved in glacier ice, providing insights into viral evolution and climate history. Guliar Glacier viruses. Ice cores from the Guliar Glacier on the Tibetan Plateau have revealed the genomes of 1,705 virus species, spanning 41,000 years. These viruses show how viral communities have adapted to different climatic conditions. 23. 3. Neanderthal viruses. Genetic material from ancient viruses has been found in Neanderthal remains, offering clues about the viral infections that affected early humans. Adenovirus, herpes virus, and papillomavirus. These viruses, which are still prevalent in humans today, were discovered in 50,000-year-old Neanderthal remains. 4. Ancient viral genomes in archaeological remains. Studies of ancient human remains have uncovered viral DNA, shedding light on historical viral infections. Epstein-Barr virus, EBV, detected in a 5,700-year-old birch pitch, reflecting the ancient human oral environment. 5. Endogenous viral elements in other species. Ancient viral genomes are not limited to humans, they are also found in other species. Koala retrovirus, KORV, an endogenous retrovirus found in koalas, which is still actively integrating into their genome. 6. Ancient viral genomes in plants. Viral DNA has also been discovered in ancient plant remains, providing insights into plant-virus interactions over millennia. Ancient Gemini viruses, detected in 1,000-year-old plant remains. These viruses have helped researchers understand the evolution of plant pathogens. 7. Implications for modern science. Studying ancient viral genomes helps scientists understand how viruses have evolved and adapted over time. This knowledge can inform the development of antiviral therapies and vaccines, as well as provide insights into the coevolution of viruses and their hosts. That was all. Goodbye.